Okay, so in the last video we were done till the introduction of enzymes. In today's video we are going to see cofactor. First thing, cofactor are the non-protein, which means they does not have protein, and they like it's not important that every enzyme will be having it particular or every enzyme will be having its own cofactor. Not not surely or not confirmed or not important. Every enzyme have cofactor. Okay, and we I just want to tell you we have two types. Okay, so the two types which we have of the cofactor. So what are that? First one is holoenzyme, and second one we have is apoenzyme. So now what are these both? First have a look to holoenzyme, which is simply like which requires cofactor. Require cofactor. For what? Now cofactor is required like the different, like we are just discussing the differentiate between these both. So holoenzyme is simply which require cofactor for what? For their activation. Like if you've just taken one enzyme or like anything in, yeah, there could be any enzyme you've just taken, and for their activation we suppose that we need to have a holoenzyme or the cofactor to activate that. This means the requirement or they require uh, they require cofactor for their activation, and apoenzyme is simply like the, if you just compare, so this means if holoenzyme does have, the apoenzyme won't be having, so cofactor is missing. This means for the activation no cofactor is required, but this will like still this will work, but will not need any kind of cofactor. And then we have, if I just tell you an example of holoenzyme, so now like we are looking into the example of these. Okay, so the example of holoenzymes is pepsinogen, which is like present in our stomach, that is in the inactive form. Like simply pepsinogen is in the stomach, and holoenzyme are like they required cofactor, and this will wait pepsinogen, wait let me add dot thought. If the pepsinogen, that is the example of holoenzyme, but when it turns to the, like when it become active, so that is known as apoenzyme, the activation. When it become active, that is known as the apoenzyme. So holoenzyme was when there is requirement of cofactor for the activation. So if I just taken pepsinogen, when it will activate without any cofactor, that would be the example of apoenzyme. And we can also say that apoenzyme is like this will not work lonely, or not will work only when it have, like this does not work separately. This requires something which would help this to work, and these have the polypeptide chain between. And we also call pepsin is an enzyme which is secreted by our stomach in the inactive form, and that inactive form would be pepsinogen. Now pepsinogen, as I just told you example, this is an inactive just because of the polypeptide chain they have, and they are known as the active site, or we can call active site to the group.